Hello and welcome to the IAS Towards an HIV Cure session, session 4, Pathways to an HIV Cure, Tools for Community and Clinicians for the AIDS 2020 virtual conference. We've got two great speakers with us today, Sharon Lewin from Australia and John Frater from Oxford. I'll introduce Sharon first and then John afterwards to take part in the debate, gene therapy versus immunotherapy, which is more likely to work. So, introducing Sharon. Sharon Lewin is the inaugural director of the Peter Doherty Institution of Infection and Immunity, Professor of Infectious Diseases, the University of Melbourne, consultant in infectious diseases physician, Alfred Hospital and Royal Melbourne Hospital, and is a National Health and Medical Research Council practitioner fellow. She is an infectious disease physician and basic scientist. Her research focuses on understanding why HIV persists in treatment and developing clinical trials aimed at ultimately finding a cure for HIV infection. She has also had a long-standing interest in the natural history and management of HIV hepatitis B co-infection. She is an elected member of the Governing Council of the International AIDS Society representing the Asia-Pacific region, and she co-chairs the IAS Global Advisory Board for the Towards an HIV Cure initiative. So, over to you, Sharon.
Thanks very much, Damian, and what a great pleasure to be debating John Frater today. I have to say, when I saw John take on Steve Deeks last year in Mexico and totally demolish him, I thought, I'm going to rise to the challenge this year. And I'm going to convince you about why gene therapy is more likely to work and finding an HIV cure than immunotherapy. And I have 5 major points to make. First of all, gene therapy is more likely to work for an HIV cure because we're ultimately after eradication, not remission. Gene therapy is more likely to work for an HIV cure because we already have proof of concept that CCR5 elimination leads to a cure. We have multiple candidates that are targets for gene therapy, each with a highly justified rationale. We have the potential for the ease of delivery, essentially one day, a one-shot cure. And John's going to try and convince you that gene therapy is plagued by complexity, toxicity and cost, but I'm going to tell you that these are improving all the time. So the overarching goal of gene therapy is eradication. This is the major barrier to an HIV cure, the persistence of long-lived latently infected cells. The ultimate goal that we need is to eliminate all those infected cells or eradication, and that's the target of gene therapy. Alternatively, we can reduce those infected cells, boost immune control, and induce remission, which is the overall goal of immunotherapy. And for multiple reasons, we need eradication for an HIV cure, not remission. The major reason we need an eradication, not remission, is to get maximal public health impact. Remission will mean that we'll need ongoing viral load monitoring. This will add significantly to cost and, most importantly, the risk of unexpected viral rebound and therefore increased risk of transmission, always a concern with immunotherapy. At a population level, we also need to have the greatest impact, and that means a cure that has a low risk of relapse, likely with gene therapy, is available to those who can't access or tolerate ART, and potentially also protects from reinfection, something that gene therapy could potentially do. Now, the most important reason why I think gene therapy is more likely to get to a cure that works is the proof of concept that CCR5 elimination leads to a cure. We have evidence of 2 effective cures, Timothy Brown and Adam Costelao, the London patient, effectively and completely cured by elimination of CCR5. Proof of concept this can work. Proof of concept that immunotherapy leads to a cure, well, that's a big fat zero at the moment. A few monkeys have been potentially cured or under long-term control, but no evidence of eradication from immunotherapy. Gene therapy also has multiple targets, which also increases my position that we're most likely to reach a cure. Gene therapy can protect from HIV, and that's essentially engineering uninfected cells to be resistant to HIV, what we're doing with CCR5 gene therapy. But gene therapy can also be used to attack or enhance anti-HIV immune responses through either T cells or antibodies. And finally, gene therapy can be used to purge or directly eliminate the virus itself. So there's 3 different approaches that are going to be evaluated for gene therapy. One of the biggest challenges that people often point out is that the delivery of gene therapy is complex. It can be ex vivo, meaning gene editing outside of cells outside the body, or in vivo, gene editing in the body. Now, ex vivo gene therapy requires removal of cells, modification ex vivo, reinfusion of those modified cells, and this has already been done. We know this is possible for ex vivo modification, for example, of CCR5, or ex vivo modification to generate CAR T cells or rebuild someone's immune system. Now, this is certainly complex and needs quite high-tech approaches to deliver it. But really, the long-term pathway of gene therapy is to develop in vivo gene therapy, where one could conceivably use nanoparticles or a vector such as adeno-associated virus to deliver the gene of interest. And this is not science fiction. In fact, this has already been done for HIV, where you can deliver a gene such as an antibody and induce ongoing, sustained, long-term production of a neutralising antibody, reported just earlier this year for the delivery of an antibody VRC07. So there are multiple delivery systems, and some of these could in fact be very simple to deliver. Finally, there are promising results of gene therapy in multiple animal models using multiple different targets. For example, targeting HIV has been proven to be effective with gene therapy in humanised mice. Delivering CAR T cells or modified T cells so they can kill HIV-infected cells, again been shown to be effective in a mouse model, or adeno-associated virus delivery of anti-HIV monoclonal antibodies, leading to long-term virological suppression of at least one monkey, but now shown to be possible in vivo as well. Finally, people always criticise gene therapy as never being practical because of complexity, toxicity and cost, but these are improving all the time. This is Jennifer Adair, who's literally invented a way to do gene therapy in a box, a machine that has about a 5-foot diameter footprint and dramatically reducing the cost and increasing potential availability. There are also far better ways to eliminate a person's hemopoietic stem cell, or what we call pre-stem cell conditioning, using antibody drug candidates rather than invasive chemotherapy or radiotherapy, which I imagine in the future will significantly improve safety. So in summary, gene therapy is more likely to work for an HIV cure. Most importantly, its goal is eradication, not remission, and eradication is what we need. We know it works, at least proof of concept for CCR5 elimination. There are multiple candidates, the virus itself, protecting cells or increasing immunity. The ease of delivery is getting better and eventually could literally be a one-shot cure. And the issue of complexity, toxicity and cost is improving all the time. Thanks very much.
And now for our second speaker, John Frater. John is an MRC Senior Clinical Fellow and Professor of Infectious Diseases at the University of Oxford. He is the scientific lead and co-founder of Cherub, an NIHR-supported UK collaboration dedicated to finding a cure for HIV infection. He also works as an honorary consultant physician in HIV and infectious diseases. The aim of his research is to understand better the reservoir of latent HIV infection in treated individuals and to pursue strategies for curing HIV infection with a specific interest in primary HIV infection. Over to you, John.
Okay, thank you very much, Damian, and for that very kind introduction, and also to the IAS for this invitation to take part in the debate. It's a real honour to be debating against Sharon Lewin, and slightly intimidating as well, but I shall do my best It's a big question, immunotherapy versus gene therapy. And if I move my slides forward, we can hopefully... Okay, I want to start this debate by just exploring a very fundamental principle that has to set the scene for everything we're going to be talking about. And this is that in the scientific field of HIV cure, there is one fundamental truth. And that truth is that Sharon Lewin is queen and she is always right. And in that context, it is very important for me to realise that there is only going to be one outcome to this debate. But I aim to do my best and I aim to go down fighting. So, what do we mean actually by immunotherapy in this context? Because it's a pretty broad thing. And I think in terms of what we're going to be saying, we're talking about a drug that generally enhances, but modulates is the term that we often use, the immune system. It makes it work better. Sometimes it calms it down, but really the idea is to make an immune system that isn't working as well as it could be work a bit better. There are many examples already. Checkpoint inhibitors you might have heard of, such as anti-PD-1 in cancer. Cytokines that have been used, for example, interferons in hepatitis C, adjuvants such as the new TLR agonists that you'll have heard about that have been also used for a while in HPV, and then more recently other sort of immune-based therapies like CAR T cells, which do overlap to some extent with gene therapy, and then direct immunotherapies like the BNABs we've been hearing about in HIV, which are really exciting. So let us just go back and have a think about what this question actually is. Here we go, immunotherapy versus gene therapy. What is more likely to work? And if we want to take this at face value, the question is actually already answered. Because as Sharon has just told you very eloquently, gene therapy wins hands down. You know, there are already 2 people cured, possibly 3 in Dusseldorf, and these cures are there and they are documented. Now, I refer you to my very earlier point, which is that Sharon is always right, and she is in this case. But what I want to do is just explore what we really mean by this question, what we mean by what is more likely to work, because I think it is a little bit more complicated than we're assuming it might be. Because what we're looking for, we could argue, is something that will fulfill these 3 criteria. It has to be able to be used on thousands, even millions of people. It has to be safe, and it has to allow people to stop their ARVs and stay well. And I think there's debate here about what we mean by cure really in the long run. Because actually, is it enough to come off your drugs, stay well, not transmit, and have the confidence that you can do that? And I could argue that actually that is potentially enough as we move forward in order to sort of define what a cure might cover. So, as much as I hate to say it and disagree with Sharon, in this scenario, immunotherapy wins hands down because it has already been given to thousands of people in different contexts. Anti-PD-1 for melanoma, it works really well. It changes lives. TNF therapy for rheumatoid arthritis has changed the lives of people living with this condition. And we're starting to hear more about these in more complex and fascinating conditions like CAR T cells, which have had FDA approval for the treatment of difficult lymphomas and leukemias. So these drugs are out there. They've been given to thousands of people and they are changing lives. They're doing amazing things. And in that context, the second point is there are side effects. Yes, some of them are severe. Some people can't take these drugs. Some have to stop them. But overall, I think one would agree that these would be considered as safe interventions. But the third question I hear you say, is it going to work for HIV? And this is the crux of it all. Because at the moment, as Sharon very eloquently pointed out, we don't know. Because if we did, we wouldn't be having this debate in the first place. But I want to take you back to a fundamental principle, which is the heart of all of this. Because our immune system is amazing. And when it works well, it is incredibly good at its job, and it can control HIV. We've heard of elite controllers, post-treatment controllers, long-term progressors. One would argue that it is the immune system in these unusual cases, but there are plenty of them, that is doing the job. And it is immunity. It is the best machine that one could imagine. It has had the hardest R&D development you can think of, millions of years of natural selection. If it doesn't work, you die. So the immune system has been finely tuned and honed over millions of years to do an amazing job, and it is amazing, and we should use it and make the most of it. And in that context, the question is, why doesn't it work? And that's what we're starting to learn, and that's where these drugs come in. Because when the immune system gets tired or exhausted, we now have the potential to up its game. We can improve on nature, which is an amazing thing to think about, and that is what these drugs do. And in the context of what is out there, there are things that are brewing that look really interesting. The broadening neutralizing antibodies that are out there. The data are remarkable. They hint at something that is tractable, lasting, and effective. And we cannot wait for the COVID crisis to end so we can really test what these drugs can achieve. But more work is needed. But what we have out there is tantalizing. So just for an example, and many of you might have seen this data before, this is from when these BNAbs were given in humans in the paper called Mendoza paper. And what these individuals had, they were given BNAbs and they stopped their therapy. And this red circle down here shows that in over 25 weeks after stopping the treatment, a number of these individuals were still undetectable compared to that dotted black line you can see, which is the control arm all rebounding. And in the animal models that Sharon also mentioned, we have animals that were given BNAbs and a TLR7 agonist, or more immunotherapy. And here, those 5 black dots, these are animals. If you just cast your eye across to the y-axis, 200 days after stopping antiretroviral therapy, these animals still hadn't rebounded. They may well have been cured. So more is needed, but it is very exciting data. And one, just as an aside, do not confuse the failure of vaccines, which I thought Sharon was going to point out, but she didn't, but I'm going to say it anyway. The failure of vaccines so far is a red flag for immunotherapy because they're being asked to do very different things. We don't have a vaccine, but a vaccine is being asked to target a rapidly mutating, quickly replicating virus. It is a moving target. For immunotherapy to work, it's very different. It needs to target an antigen that is stable and conserved. And in terms of the HIV reservoir, we may well have that to a much better degree. It's not perfect, but it's better. And if we think of it more like cancer than infection, we have an antigen that is stable that we can target. And we know that immunotherapies can work really well for cancer. So that is a good argument to start from. Okay, to sum up, here's a table that sums up, I think, everything. Gene therapy versus immunotherapy. Experience with thousands, not for gene therapy, and you're not going to get it. Immunotherapy, it's there already. Is it safe? You know, with gene therapy, we don't know. At the heart of it, we don't really know how our genes properly work yet, if we're going to be really honest with ourselves. So messing about with them is still likely to be risky. With immunotherapy, we have got a much better understanding of whether these drugs are safe or not, and I think we can move forward. Does it work? Yes, for gene therapy it can, but not in everybody. For immunotherapy, well, we don't know, but we do know that your immune system is amazing, and that has to be a platform to work from. So as an analogy to end on, which horse would you invest in? Would you take the stallion, who's had a couple of really good results, but it's just a bit knackered and it's a bit inconsistent, and that's your gene therapy, it's not the future? Or would you invest in this animal, the young pedigree foal from an amazing stable, from winning stock, but hasn't got any races under its belt quite yet? And my argument is this is the way to go. So I will stop there. Thank you very much.
So thank you very much for that, John. Now, as usual, we would kind of go to the floor to take questions, but as this is a recorded thing, we've had to get some questions from community members. So we're just going to fire some questions that we've received from various people around the world to both John and Sharon. And I'll start with Sharon, I think. How do immune system modulators work against immune modulating agents?
I might just get you to repeat the question, actually, Damian, if that's okay. How do immune modulators work?
How do immune system modulators work against immune modulating agents?
I think the question's asking how does immune modulation work, perhaps, and John gave us a nice summary of the ways that these can work. I like the analogy, which actually comes from Marla Maini on immunotherapy for hepatitis B, and she talks about immune modulations revving up what's already there, so things like TLR agonists or interferons, for example, so turning on the immune system, releasing the brakes on the immune system, so it's, you know, how you release the brakes so you can go faster in the car, which is immune checkpoint blockers, so reversing an exhausted immune system, or replacing the whole car with a new immune system, like using CAR T cells. So all of these approaches are addressed at trying to address defects in the immune system, and some are a gentle revving up of the immune system, some reversing the brakes, and some are replacing the immune system altogether. And added to that, immune modulation can be HIV specific. So John gave the example of BNAMs are broadly neutralizing antibodies. They're just targeting the HIV virus. Or they can be non-specific. So immune checkpoint, for example, has released the brakes on all cells. Therefore, you activate your HIV-specific cells, but also other parts of the immune system. And that's the sort of framework that I often think about for immunotherapy.
Okay, thank you. John, you touched on some things having side effects earlier on. So there's a question here. What are the side effects of immune system modulators?
Potentially lots, and I think one has to be realistic about that. I think, the immune system is a very powerful organ, if you like, as part one of the sort of fundamental structures of our bodies. And when we feel ill with flu or infections, often the reason we feel awful is not because of the bug itself, it's because of the way the immune system is producing its response. So for many of these immune system modulators, the side effects that you see can include kind of the sort of flu-like symptoms, feeling quite unwell, fevers, other things like that. And they can be severe in some people. And many of the people can get through those, and that's fine, but there are more severe side effects. So one of the dangers of ramping up the immune system is it starts to target you. It starts to think that you're foreign, that you're the target. So, for example, the immune system can target the lungs. You get something called pneumonitis, or it might target the bowels. You get something called colitis, or the liver, something called hepatitis. So if the immune system gets over-ramped up, if the foot presses down the gas pedal too hard, then you're in trouble. And there are plenty of cases of those being side effects. And so one has to be careful. But I'd argue that if that's also a measure, these side effects are also a measure of how powerful this is, as a potential therapy. So in the realms of this debate, the immune system can do amazing things. So we almost need to kind of hold it back sometimes to make sure it doesn't do too much.
Okay. Thank you. I'm just looking at the time, and I'd like to get two quick questions in, if possible. Sharon, one is about reinfection, and will either of the options proposed prevent a reinfection from occurring?
Yeah, John could have gone to town on this one. He spared me on it. Actually, he was very gentle. He was very nice. He was much meaner to Steve. We often think about cure as eliminating the reservoir, but actually protecting against reinfection, I touched on it, is in fact really important for having a public health impact. And many of our strategies are not directed to protection from reinfection, but gene therapy can do it, and maybe immunotherapy can do it. So gene therapy, if you're CCR5 negative by gene therapy, you're effectively protected from reinfection from most strains of HIV. Of course, there are some strains of HIV that don't use CCR5. So you're 98% protected. We all actually know that Timothy Brown's actually on PrEP because he's not protected from some strains of HIV. So it's partially protective. But if you have a really good immune response that you use to eliminate the reservoir, it could technically protect you from reinfection. So CAR T cells, John touched on them. I grabbed them for gene therapy. He grabbed them for immunotherapy. CAR T cells are really a form of gene therapy because you take out the T cells, you modify them so they recognize HIV, and you put them back in a person. So I'm grabbing them for gene therapy. CAR T cells, which are being used for cancer, actually hang around for a long time. So they potentially could protect from reinfection. And then some of the other approaches, again, I talked about antibodies that can be delivered via gene therapy, much more effective, I should say, than how John wants to deliver them by regular injections. But antibodies could also technically protect you from reinfection as well. So, both sides, that could be a potential benefit. I should say, be serious, that in most, we just want to get to first base, which means allowing people to stop ART. But really, for the long haul, ideally, you need this to be protected as well.
Yeah, absolutely. So we're coming into the last minute and a half, but I think this is a really important question, and it's particularly focused around women and girls. We know that women, for many reasons, are obviously quite often left out of clinical trials. So I think if we look at both the options here and look at girls and women, which is the best and most suitable option for this population? And is there a difference between premenopausal and postmenopausal for the options on the table currently? I will let either of you pick this question up.
I'm being pointed, and I'll try and answer. I will just say before I answer that question, just in response to Sharon's little dig there, the two best examples she gave for her gene therapy were these CAR T cells and the antibody-mediated delivery of the AAVs. They're both immune system function. It is the immune system you're getting your gene therapy to make work better. So at the end of the day, it's the immune system. But okay, getting back to your question, I think that's so difficult, but it is important. I think one of the things we would say, we don't have a lot of platform to work from. I think what we know from the immune therapies that are out there so far and how they've been applied to different conditions, they're not fussy about gender. They're working well across those. I think in terms of age, for younger people, one might be a bit more nervous about gene therapy in a younger person, because you could argue that actually we don't really understand how manipulating genes work, especially if someone is developing. So but these are great questions that are so important, but actually, we don't know the answers yet. But I think from the immune side, I think there is no real reason to feel you wouldn't be able to use them for everybody.
Okay. I think that these questions are really important, and some of these questions have come from girls and young women living with HIV who feel excluded from trials. And I've been asked to point out that more than 50% of people living with HIV are women. So just when we're moving forward in clinical trials and moving things into trial settings, to really try and include the women and girls as much as possible in a safe way, obviously.
And just to add to that, Damian, I think what we're seeing in a lot of the post-treatment control studies, there is definitely a difference in that women are doing better. So there's more argument there for making sure we do include women in these studies as much as possible.
Yeah. I've got a full list of questions here, but we are actually out of time. So I think maybe if we can get these questions to IAS, and IAS will put them up on a website somewhere with the answers. So I'd like to say a very big thank you to both Sharon and to John. Sharon, I know it's very late at night down in Australia. John, it's the middle of the day here on the hottest day of the year. But thank you both very much, and thank you to IAS for this session. Take care, everybody, and keep safe.